when John called me, he said this was my task. I changed it a little bit, and we discussed this over the phone. I said I would say discuss cure when possible, conversion to a chronic disease when cure is not possible. And I think actually we're close to getting there, but we still have a lot to do. And so I'm going to use several examples of what we could do now, what is in the near future, et cetera. I'll cover 4 areas. One will be trying to maximize cure with current drugs. And some of the strategies I think are sort of shovel ready, using the old Obama term. And so I'll use I'll use bladder cancer as an example. This is from the meta-analysis for neoadjuvant chemotherapy. You'll see that there are 2 curves here. I think we've got a pointer here. Yeah. So we've got 2 curves, and one is cystectomy alone, and the other one is neoadjuvant chemotherapy followed by cystectomy. In the meta-analysis, hazard ratio is very good in terms of 0.86, so it is a benefit, level 1 evidence, standard of care, but a minimal actually number of patients really benefit. And there have been multiple studies showing that patients who respond well to chemotherapy, as evidenced by the pathologic assessment at the time of cystectomy, actually do well. So these are patients who respond with no more in the way of muscle invasive disease, we'll just say less than PT2, and they do well. Patients who do not respond, we lump them together here, greater than T2 or greater, do less well, and this has been well published in multiple series. But it gives us the opportunity to be able to examine extreme responders, the responder versus the non-responder. And this gets into the shovel-ready type of approaches that we can look at. So this is from Ellie Van Allen and Jonathan Rosenberg, and they did exactly that. They look at the responders versus non-responders, as we just defined, and examined whole genome sequencing to see if there are any clues about which patients are more likely to respond. And in fact, this is some of the sequence data, and you can see that there are a number of mutations here, including TP53, RB, ARID1A, KDM6A, not really informative in any way in terms of a difference. But then you see ERCC2 here. These are the responders in the blue. These are the non-responders in the yellow. And you see all these patients with ERCC2 mutations, a clue that response is potentially predetermined. So this is straight out of the manuscript. ERCC2 demonstrates statistical significance with regard to response, and you'll see that all the ERCC2 mutants are on the responding side, and none of the patients who didn't respond had mutant disease. They examined the actual areas of mutation. They're in conserved areas. They're very similar to what has been found in 2 other databases, the Chinese database and the USA TCGA database, in terms of their location. And in terms of TCGA, it's about 12% of tumors have 12 to 13% have ERCC2 mutations. And then there's a few of other diseases. So obviously it needs to be validated in a larger set. Jonathan has a grant to be able to do that, but it's a first clue that using current sequencing techniques, we might be able to identify patients who are likely to respond right from the beginning. We also have a second data set. It's a little bit different, but the same kind of concept. This is from Betsy Plimick, looking at the MD Anderson and the UPenn data. Same kind of issue, and that is looking at several genes that are important in terms of DNA repair, ATM, FANC-C. RB1 data is a little bit different than what you saw from the previous study, but these are the patients who have mutations in one or more of these genes, and you'll see that they do quite well. And so that issue of cure, issue of chemotherapy sensitivity needs to be validated in a larger data set. And in point of fact, actually, it provides a great opportunity for large-scale study in terms of next-generation sequencing to be able to identify DNA signatures. It's not just in the urothelial cancer. There's a study, also an extreme responder, but looking at one patient. There's a patient who had small cell of the bladder. This patient had been treated with etoposide and cisplatin, responded but relapsed here, underwent a nephrectomy and node dissection, relapsed again, and went on an experimental drug, AZD, along with irinotecan, and had a remarkable response and is still NED years later. This patient also underwent whole genome sequencing, published by a group. You can see it's a big time effort in terms of coverage and in terms of candidates. You see 19,000 candidates, but boiling it down in terms of a mutation that has a high likelihood of association with response, and it was in the RAD50 complex and responsible for the hydrolysis of ATP in that molecule in RAD50, and that collectively with MRE11 is responsible for regulating ATM and DNA repair. And so this patient treated with irinotecan because the patient had a clonal disease, the RAD50 mutation, treated simultaneously with this JAK inhibitor, is a complete responder. So it gives you an idea of the power of sequencing, and should be something that we'd be doing on a regular basis to try to exploit some of these observations, and also in terms of new drug delivery. So a second area is characterizing molecular alterations of novel drugs. This is just not happening across the board in terms of GU cancers. So John mentioned the meetings down in Puerto Rico, I believe it was, for the thoracic meetings. If we were having that meeting now, this is what our thoracic colleagues do. All patients get sequenced. They know the mutations that are tractable. Patients are treated. And in fact, if you take a look, even in the ALK rearrangements, it's only 8% of tumors, but they have 2 drugs in terms of approved drugs in terms of treatment. And we should really be doing this in our diseases in terms of what we have. That's been studied in urothelial cancer, and we have it in other cancers as well. We know that we have a fairly, at least in bladder cancer here, we have a fairly large and extensive list of alterations in a number of different pathways, many of which are actionable, some of which are not. So histone modifications are not. But in terms of pathways here and here, and I'll just bring this up a little bit more. In the p53-RB pathway, we've got potential actionable areas. Same thing with regard to the RTK-RAS PI3 kinase pathway. And in fact, all of these are potentially actionable. And we really should be along the same path as our colleagues in lung cancer. But in point of fact, there's a catch-22. Patients can't necessarily get sequencing for their tumor because it's not covered by insurance, has to be on protocol, has to be CLIA certified to be able to make clinical decisions, or it's a companion diagnostic, or the company may not want to pay for screening for the patients for an 8% mutation for their particular drug. So really the barriers here are substantial. And so we're into this catch-22 that we do have drugs, and some of them are in study right now. Accrual is slow, and it's unfortunate because we really could accelerate treatment if we had widespread sequencing. So that brings up the issue of new ways of addressing these kinds of deficiencies. So BCAN is the Bladder Cancer Advocacy Network. Many of the people in this room have been affiliated with them, and they have actually identified a donor who has submitted or provided funding for patients to be sequenced to make them eligible for clinical trials. And it's this kind of novel interactions that I think will be important as we move forward, because we won't necessarily have the funding from our federal colleagues as much as we would like. So that's a challenge for us. Now, sequencing with regard to novel drugs is important because we already have identification that patients can respond exceptionally well. So this is a patient who was treated on everolimus, an mTOR inhibitor. There were 45 patients in this trial, no sequencing done up front. This patient is an extreme responder, 1 CR out of the 45 patients. She underwent treatment after progression of disease with standard chemotherapy, with gemcitabine and cisplatin, and received everolimus and responded nicely. She underwent whole genome sequencing. As you can see here, a fair number of somatic mutations, 140 coding mutations. 2 mutations were particularly important in terms of rendering this patient's tumor exquisitely sensitive to everolimus, and she is still NED for 5 years. We just saw her last month in the clinic and doing exceptionally well. So it shows you the power of this type of approach in our diseases as well. The third area I'd like to discuss actually is checkpoint blockade. And Obviously, this is an area of great interest to all of us. And I just want to cover the issue of characterizing response. And because we talk about cure and we talk about turning an acute disease into a chronic disease. So what are some of the considerations here? So to really look at immune therapy, allow us to take a step back. It's actually been going on for a long time. William Coley actually was from the hospital, which is now Memorial Sloan Kettering Cancer Center. He was a young surgeon. He actually observed that patients with sarcoma did better if they had had a history of having a superficial erysipelas streptococcal infection. And so their outcome was better after surgery than patients who had not had an infection. So this is remarkable. Coley later combined a gram-positive heat-killed streptococcus with a gram-negative heat-killed serratia, made a vaccine, and administered it to hundreds of patients. And he did this in the 1800s. Actually, publications first started in 1899. And so in the middle, you see this picture here of William Coley and his two young colleagues, Walter Stadler on the left and Oliver Sauter on the right, in their younger days, working diligently with young William Coley. Chuck, I didn't include you, but I don't think it's a problem here, right? And so this is the 1899 publication, and it's a sarcoma. This patient rendered free of disease after an erysipelas infection, and actually the follow-up is 7 years. Actually, if you look at the bottom, 7 years after his treatment, and so it's remarkable when you think about it. I'm actually going to come back to that in a moment. And so all of us have seen this, the new immune therapy, the PD-1 pathway, and all the exploitation. And we know that this year, I think the pharmaceutical industry will spend approximately $4 billion, I think, in terms of immune therapy across all diseases. And I don't have to go through the mechanisms, et cetera, in the different, PD-1 and PD-L1. We've all been through this with regard to checkpoint blockade, and we're starting to see the studies. I'm actually going to focus on a couple of aspects of these that are pertinent to GU, areas that we might be able to exploit. So the first thing is we have the candidates, in terms of somatic mutations. The dogma is the higher the number of mutations, the greater the number of neoantigens, the more likely they are to respond. And we have that here. We know that drugs have been approved in melanoma and non-small cell lung cancer, and bladder's right behind because of the issue of the high number of mutations seen in that disease. I'm also going to come back to this in a moment. And we've seen 2 studies that have been published already, and actually we're seeing follow-up as well. But we've got atezolizumab that was studied, actually in a phase 1 trial with an extended cohort with urothelial cancer. And atezolizumab, the study included all patients, irrespective of the staining of PD-1 and the status of the tumor, and then broke it down retrospectively in terms of response. And I'm not going to go into all the controversies about PD-1 expression and what antibodies use and the conditions and how you read it, et cetera, because it's different for every companion diagnostic. But I am going to cover a couple issues of this issue of cure and chronic disease. So this comes from the original cohort that was presented with regard to response and survival. And the issue here is that tumors that had already been recognized by the immune system and having a checkpoint blockade, the response rate was quite high. And those tumors had little or no recognition, at least by this assay, archival specimens with all the conditions, less in the way of a response rate. And the take-home message is that PD-L1 positive tumors respond better. I don't think that's the important issue. I think this is the important observation when we talk about cure or turning an acute disease into a chronic disease. So this is the swimmer's plot, and I'm going to show you several of these. This is a swimmer's plot from that publication, and this is 1 year. And if you take a look at all the phase 2 studies that have been conducted in this particular setting, previously treated patients, the median survival is basically 7 to 9 months, and it's a very reproducible number. On the other hand, you see all these patients with these arrows who are still responding well over a year, turning an acutely ill and potentially deadly disease into a chronic disease, and some of them with substantial duration, and that's still ongoing. So then we have pembrolizumab. Now it's anti-PD-1 instead of anti-PD-L1, this is all patients irrespective of their, obviously all patients who have PD-L1 positivity, different assay, different reads, et cetera. But the important aspect is the same kind of observation, that a number of these patients have reduction in disease, as we can see, and about 2/3 of these patients, selected based on their staining characteristics, show that they respond with the same swimmer's plot. And that's really the important observation. Smaller number of patients, a smaller study, but here we are at 60 weeks. We're well beyond a year here, and we have these arrows that are continuing in terms of continuing response in immunotherapy, something we've never seen before in patients with chemotherapy for this disease. It doesn't stop there. So these are data that were presented by Pam Sharma a couple of weeks ago in Vienna, looking at Checkmate 025. It's a randomized trial in renal cell carcinoma, all previously treated patients in terms of prior anti-angiogenic therapies. Patients are randomized to nivolumab versus everolimus, a standard of care for second-line therapy, straight 1-to-1 randomization. I'm not going to go over all the data because that's going to be discussed in the renal session. I'm just going to cover a couple aspects. One, we're more response rates, a 25% response rate versus a 6. But I do want to draw your attention here when we take a look at the number of weeks here, 64 weeks. We draw a line up here. Look at all these arrows, these continuing responding patients, that continued response that we see with immunotherapy. And that's obviously an important observation in terms of if you can't cure, you can extend survival. And this is the survival. And the only reason I'm showing these survival distributions is this is everolimus, the standard of care. This is nivolumab. This is the median survival of 25 months for second-line therapy. But let's go back 10 years on this disease. Let's go back to 2005 at the ASCO GU session in 2005. And this was a 4-arm trial for first-line therapy. And it included progesterone, megase, interferon, interleukin-2. This is a sub-Q interleukin-2, and interferon plus interleukin-2. And these survival curves are absolutely superimposable. First-line therapy with median survivals of 15 months. Now we're seeing median survivals of over 2 years as second-line therapy after drugs like pazopanib and sunitinib. So this is what we are hoping for over the next decade. So we're back to the somatic mutational frequencies. We have the two diseases. What have they learned that we can also tag along with regard to our studies? Well, the first thing is mutational burden seems to play a role in melanoma. And so this is a nice, elegant study from Alex Snyder in Tim Chan's lab. A pretty straightforward analysis looking at mutational load. And these are patients who have long-term benefit, those who do not, and the mutational, non-synonymous mutational load is higher in patients who have a benefit. She did it in two different data sets, a discovery and a validation data set, and you look at the patients who have greater than 100 mutations versus those that are less, and you see a splay of the two curves, and mutational load seems to make a difference with regard to melanoma. And the same thing has been done and reported by Naya Rizvi, same group, Tim Chan's lab, and this is the same methodology, a discovery group, a validation group, high synonymous mutation rate in non-small cell lung cancer versus for those who have derived clinical benefit versus those who do not have benefit. There's a discovery cohort, a validation cohort. Let's just take a look at all tumors on the bottom right, and you see that the patients with a high mutational load do better. They've taken it further in terms of mutational signatures. And this is from the non-small cell lung cancer. They actually defined a smoking signature and you can see patients whose tumors had the smoking signature did much better than those who did not. So now we're into companion diagnostics with regard to predicting likelihood of response. The melanoma one is actually, I think, more interesting, and that is what Alex and her colleagues did is they looked at neoepitopes that were associated with response, both in the patients who in discovery and validation set, and they find these neoepitopes, and patients who have this signature do much better in terms of survival versus the patients who do not have the signature. And they go on further and they analyze the neoepitopes, in analysis of the neoepitopes, they look at tetrapeptides. And these conserved tetrapeptide neoepitopes actually comprise substrings of antigens from infectious pathogens. Now, these are predicted antigens, but you can see that those that are in the benefit group versus the non-benefit group, that there's obviously a difference with regard to these tetrapeptides, suggesting maybe Bill Coley was onto something here, that maybe there has been recognition in terms of previously existing antigens, and now you're seeing recognition in melanoma, potentially. Now, these are predicted, and the difference between predicted and actual is something that we need to work on with regard to these various diseases. So we're back to the somatic mutation rate, and we have melanoma, non-small cell lung cancer, bladder seems to follow on, but then we have renal cell carcinoma. It doesn't really fit the mold. And in fact, in renal cell carcinoma, the time to response is different, whether or not it has no relationship to PD-L1 expression, at least so far with nivolumab. And then the prostate is a completely different animal as well. And they have mutation rates pretty simple, at least median and mean mutation rates pretty similar. But are there other clues? And this is a nice study, actually, from the Goldstein group. And they looked at colon cancer. It's an interesting story because in their first study in terms of colon cancer, only 1 of 33 patients actually responded. And so they looked at that patient and said, well, maybe it's a high mutational rate. So what they did is they looked at mismatch repair genes. They figured this patient probably has a mismatch repair gene. They have a high associated mutational rate. That wasn't, in fact, the case with that one patient. They went on and did this study looking at colorectal cancer, those that have impairment of the mismatch repair mechanism, and those that are proficient. And you see in this study, of the patients who had a proficient repair mechanism, there were no responses. And it was 40% in patients who had mismatch repair impairment. They further looked at the other patients who are Lynch syndrome patients, who had disease within the other areas of the GI tract, and they responded just like the Lynch syndrome colorectal patients. And so the take-home message here is that, yes, they have high mutational rates, and it really provides an area of intense study right now. So we take a look at the two survival curves on the right here. This is the colorectal, and these are the patients who have mismatch repair impairment. These are the patients who are proficient. You can see there's a marked difference. These are the patients who are non-colorectal, who are other GI malignancies within the GI tract, who do quite well. But we have those two in GU. We have them in urothelial cancer, upper tract, predominantly, but also in the lower tract. And we also have them in prostate cancer. That's been well described as well. So this is an area that we should really try to potentially exploit in terms of tumors that are more likely to respond to immunotherapy. But immunotherapy is not just more than a single checkpoint blockade drug. There's a very simplistic cartoon, but we can turn up cells by blocking the inhibiting activity on a T cell. And we've really focused on PD-1. CTL4 has also been published now, and I'm going to show you some of those data. But then we can also turn up the activating part, and there are a number of studies now looking at OX40 and GITR, et cetera. And we have the potential for multi-drug studies in terms of our diseases. So why is that important? Well, let's take a look at the publication by Larkin et al. It's a 3-arm study. It looks at combined nivolumab and ipilimumab versus monotherapy. And this is all in patients with melanoma. And so in the top panel are the patients with nivolumab, and the bottom panel are patients with single treatment with ipilimumab, and in the middle is both. And the first thing you notice is that if you look at 2-drug therapy, we have a higher, the waterfall plots really show a higher amount of response here in terms of single drug with either one of these drugs. That's the first observation. They tend to do a little bit better in terms of survival. But here is the other issue, and that is that the bottom right-hand corner, you look at the patients who are PD-L1 negative, and it looks like 2-drug therapy is beneficial irrespective of this PD-L1 staining. So maybe that PD-L1 staining that we're all doing by immunohistochemistry, hopefully it'll be a speed bump and we can move on to real science pretty soon. But what about the tumors that do not show any evidence of immune response? And so this is work actually that's going on by Dimitri Zamarin, that you can infect tumors with oncolytic viruses, and that might result in type 1 interferon production in immunologic cell death. And so you can induce lysis, et cetera. It'll turn on cytokines, and you can enhance both adaptive innate immune recognition and destruction. And so this is just one study of many, and it really is controversial. Should it be systemic? Should it be injection into the tumors, et cetera? But he's looking at Newcastle virus, and there are a whole number of viruses that can be examined. This one does not have any pre-existing immunity because it's really it affects birds and not humans. It infects the majority of cancer cells. It can induce a type 1 interferon dendritic cell maturation. So it's a good opportunity to be able to look at a virus to induce immunity. They've created or engineered a virus that's expressing co-stimulatory ligand. And here are the studies. These are in melanoma. And the blue here is just the engineered construct itself, and this is in red the engineered construct plus ipilimumab. And so this might be the area that we move forward with regard to some of the other novel treatments in harnessing the immune system. So we're closer to the North Pole here than where we usually live. And John said, well, what are we going to do in the next 10 years? And so this is my Dear Santa letter. We've been very good this year. We actually have been in terms of productivity, if you look at it. Here's our wish list from your favorite GU oncologists, radiation oncologists, surgeons, et cetera. One, we really need to escalate sequencing into our diagnosis and treatment, and applying next generation sequencing to current therapies would yield immediate dividends. I showed you that in bladder cancer. It would be a major step forward. Second, we should be looking at liberal biopsies at baseline and at progressive disease for all of these diseases. And that would provide insight both on our in mechanisms of resistance and then help us with regard to strategies. Third, inclusion of whole exome sequencing, whole genome sequencing, RNA-seq. We should be really rapidly including these as the costs de-escalate. We should be including these as much as possible, whenever possible. And the payoffs will be helpful in terms of targeted therapy, epigenetic modification, and immunotherapy. Number 4, we should really be rapidly escalating the multi-drug immunotherapy trials with available drugs. We've already seen in melanoma, they're way ahead of us, and non-small cell cancer is not far behind them. And in fact, we should also embed intensive companion diagnostics in all of these so we can really dissect the biology as we have available new agents. Number 5 is novel approaches. We really need trials that will allow us to get a rapid readout so we can support the best therapies if it turns out it's a viral construct, et cetera. And then lastly, partnerships. Partnerships old and new. Yes, we have our funding from the NIH, et cetera, and other sources, but now we have ASCO's helping sponsor trials, clinical trials for the very first time. You saw BCAN is sponsoring sequencing for patients. We have other organizations that are doing this for our other diseases, and I think we should really take advantage of that and encourage resources from other areas. So I'll close there, and I'll thank you very much for your attention, and thank you very much for the invitation, John.
I would say if there are any questions, go ahead and ask. Those are tour de force. Absolutely beautiful. Questions, comments?
You know, I like your wish list, and Santa may give it. When you look at, again, I think you're exactly right, especially with what you see, is if we could do biopsies, more biopsies and do analysis on it. The question is, again, is always kind of who will generally pay for it. But isn't this something that basically, you could say the insurance companies plus minus the federal government, through Medicare and Medicaid should be kind of requiring. So if you're going to get one of these new expensive drugs, they require you to get biopsies, because ultimately, if your curves are showing 25%, 35%, 40% survivals, they need to find those 60% that aren't benefiting. overall that's going to save them a lot of money in the long term if we could prove it to them. But that may be where you can, again, if you're going to go on this brand new drug, it's required a tissue before, tissue after. If you do full genome sequencing, what's that? $10,000 added to the cost. So what is your thoughts on, basically approaching it instead of the people developing the drugs, the people who are actually paying for the drugs to be delivered?
So I think, as we start looking at all these new drugs, and they're expensive, there are a couple observations. One is, and many of us have consulted with the FDA. The FDA's charge is not to look at money ever, okay? And I think, not everybody knows that, okay? So they're going to approve a drug based on efficacy. But downstream, no matter how we look at it, we're going to have to ration, okay? And so you can ration by just costs and co-pays, et cetera. I think we as a community should be rationing based on science, just like you're talking about. And I think the only way we can make that compelling case is to identify patients who are very unlikely to respond, and increase the denominator, or the patients, or selection of the patients who will respond. Now, the first cut, I don't think PD-1 is it, to be honest with you. Or excuse me, PD-L1, immunohistochemical staining is it. It reminds me of staining for P53 mutations back in the old days, and I choke on it. I think a lot of us do. But, we can get to this very quickly. And in fact, if you take a look at the costs in terms of, whole genome and whole exome, and actually even just, next generation sequencing, 400 genes you can do very cheaply. I think if we had signatures, or we had panels for chemotherapy sensitivity with cisplatin, if we have it for, other novel drugs, we would be rationing our resources based on science. And that's, I think, the position we have to continuously take, rather than rationing based on our insurance policy or our co-pays. So I agree. By the way, to prime the pump at our institution agreed to sequence last year and this year 12,000 patients for free, to put them on trials, to get the process going. So I think we need that commitment from other institutions, organizations, et cetera, and we need the basket trials, so that FDA is not thinking solely on the basis of histology, but thinking on the basis of biology. And I think there are many of us in the room who feel exactly the same way. I could see a lot of heads going like this. Good point, John.